All right, folks, we're going to get started. It's my pleasure to introduce Martina for our EEB seminar, Martina Del Bello. She's joining us from Yale, where she just started her lab. Most recently before that, she was at MIT with Jeff Gore, doing really cool kind of integrative community ecology work, thinking about metabolism and how resources are shaping microbial communities. So I'll let Martina finish her introduction with a cool slide showing us everywhere she's been. But we're really excited to have her here. And that's not my water. Okay. Welcome, Martina.
Thank you. Thank you, Luis, for the introduction, also for hosting me. And this has been a very nice and exciting day. I had a lot of nice chats about science, and I'm really glad that I got to know so many people in this department. And yeah, so today I'm going to talk about mostly what I did during my postdoc in the Gore lab, and it's going to be about how the environment modulates microbial interactions and community assembly. But first, yes, I'm going to try to just place myself just to show you where I've been. So I was born in Italy, in this very tiny town. So we have our own lake. For us, it's a very big lake compared to what we have here. Like nothing, it's a pond. But we have a very nice castle, and I picked a picture with the snow just because it feels like it's a good parallel. And then actually I moved to Genova for my undergrad, and then I did my PhD in Pisa in marine ecology. And maybe many of you know Pisa because there is the Leaning Tower, not really because of its university, but it's actually pretty old, 1343. So it's one of the oldest universities in Italy. And then I did a big jump. I crossed the Atlantic, and I decided to change departments, so go from marine ecology to physics, and at MIT, where I did my postdoc. And finally, I ended up slightly south compared to Boston, in New Haven, where I started 7 months ago, a tenure track position, and I'm really excited for what's to come. But okay, to start, I want you to forget the planet Earth and move to Mars. So I recently discovered that microbes might be responsible for the absence of life on Mars. So this paper that is cited below has suggested that originally on Mars, Mars was able to basically nurture some hydrogen-consuming, methane-producing microbes that were kind of thriving on Mars, but they produce so much methane that the planet cooled down to a temperature that is equivalent to minus 200 degrees Celsius. So not so cold, but very cold still. And this actually, it's what made Mars inhabitable. So, but what's striking to me is that if microbes were actually what caused the absence of life on Mars, on the Earth, they did the exact opposite. So microbes, we know, live pretty much everywhere. They inhabit every corner of the planet, and they perform crucial functions for our health and for the well-being of the planet. And so, for example, we have microbes residing in the gut that help us digest the nutrients the food that we eat and absorb nutrients, but also they are responsible basically for more studies are showing that they really control our health. But also across terrestrial and aquatic ecosystems, microbes regulate the degradation of organic matter, and they are able to cycle back important nutrients to primary producers. And also by dying in the soil and in the ocean, they contribute to the long-term storage of carbon. And across these systems, also they regulate the production and the consumption of important greenhouse gases like carbon dioxide, methane, and nitrous oxide. But importantly, it's usually not a single species that is responsible for this function, but actually functions emerge from the thousands of interactions among 1,000 species that compose these complex communities. So I would say that thanks to the advent of 16S sequencing and other omics, we've become really good at understanding who these microbes are. So basically where we see certain microbes and where we don't find certain microbes. But actually, I think we're still far behind in understanding why we see certain microbes in certain environments and not in another one. And so what the question that kind of guides my research and in the past and also in the future is trying to understand actually how the environment structures communities and trying to find, I would say, as much as I can, rules that can be generalized to try to, and to different environments and that describe basically how communities assemble and function. And I think this is becoming particularly important because many of the environmental factors that are crucial for microbial communities, like temperature, oxygen, and nutrients, are being altered by climate change. So we really need to understand how these environmental factors structure communities to predict how these functions are maintained and also probably try to think about way to prevent the collapse of these functions. Okay, so one reason why we probably struggle in understanding why certain species are where they are and not the opposite is because different approaches that people have used to study microbial communities have not really been integrated much. So for example, the community where I did my postdoc, that is the biophysics or ecological systems biology community, uses a lot these very simplified systems where you study the interaction between maybe 2 or 3 species, and most of the time one of the species is E. coli. I have to add that most of the times these species might be what you have in the freezer, so not really guided by some specific environment. And this approach has the advantage of being extremely tractable. And by doing these experiments, we learned a lot about the effect of certain environmental variables like temperatures on the outcome of species interactions. But it's pretty unclear up to now whether the results that we find in these simplified communities matter for what we see in the environment. Then there is, on the other side of the spectrum, I would say the microbiome science or environmental microbiology that takes kind of a more top-down approach. Usually we take advantage of the various omics and we directly sample, we directly kind of get the composition of environmental samples using, again, 16S sequencing and so on and so forth. And this approach has been pivotal for us to understand, really uncover what's the diversity of microbial communities on the planet. But on the other hand, we are struggling to establish causal relationships between changes in the environment and patterns in communities. So while this approach kind of retains the complexity of real microbial communities, it's pretty untractable. So the type of approach that I've been using in the past years and that my lab is going to use is kind of trying to bridge these 2 approaches. So in the first one, I'm using results that are generated by these very simplified experiments to generate hypotheses that then I can test in using the wealth of data that is becoming increasingly available thanks to sequencing techniques becoming more affordable. And the other approach that I use to merge these 2 approaches is actually adding a step in this top-down approach. So what I do, I take samples from the environment, I bring them to the lab, where I do sort of enrichment techniques, where I can actually control really tightly the environment. So I can control which nutrients, the temperature of the environment, and then I can still leverage omics techniques to, for example, learn the composition. And what this system allows is to have high replicability, but also control on environmental variables. And in this way, I can really establish causal relationships between changes in the communities and changes in the environment. So it's kind of a compromise because we are at an intermediate level of complexity that is more tractable and compared to the 2 species that I was showing you before. Okay, so the goal of today is to tell you about 2 stories and that kind of address the question that I posed at the beginning. And these 2 stories kind of represent 2 examples of the approaches that I just described. So the first one is about how available nutrients shape bacterial community diversity. And the second one is how temperature structures bacterioplankton communities. So let's start from the first one. In this project, we asked specifically how the number, type, and concentration of resources affect bacterial community diversity. And what I'm going to show you today is that we found that individually supplied resources sustain quite diverse communities, but combining these resources only results in a weak increase in community richness. And finally, that increasing the concentration of these individually supplied resources actually reduces diversity. Okay, so why I started addressing this question? Well, the relationship between, let's say, the number of available resources and the diversity of a community has been puzzling ecologists since MacArthur and Hutchinson. But it's not just that, because nutrient pools characteristics vary in space and time. So for example, let's think about the rhizosphere, that is the area around plant roots, that can sustain really rich microbial communities, and its analog in the ocean, the phycosphere, that is the area that surrounds unicellular primary producers like diatoms. So in these particular microenvironments, the primary producers are releasing many organic compounds that are quite diverse. They can be sugars, organic acids, amino acids. So really, and depending on the status or the health of the primary producer, which specific carbon source is available might change. And second, in these microenvironments, as we move away from roots or from the cell, the phytoplankton cell, nutrients become less concentrated. And so, and these changes in nutrient concentration not only happen on local spatial scales, but also along long spatial and temporal scales, like for example across seasons or across latitudes. So really, the goal of these sets of projects was really trying to vary the various characteristics of nutrient pools and try to understand how they affect diversity. So, and to study these effects, I employed the second approach that I was telling you about before. So I started from a soil sample, and here it's me on the lawn of our previous building when I was at MIT, using this fantastic high-tech Apple core to collect samples. I was in a physics department, so that's what we were provided with. And then I went back to the lab and basically tried to detach the microbial cells from the soil particles through a process of sonication. And I use this microbial suspension to inoculate these simplified, let's say, microbial microcosms. And each of these microcosms differed because of the nutrient environments. So I started from a pool of 16 carbon sources, and you can recognize some simple sugars, disaccharides, organic acids, sugar alcohols, and then there were some complex polymer and one. This is it's very frequent in root exudates, so I was trying also to provide carbon sources that kind of made sense for a soil community. And in this environment, each of the resources could be provided as a single source of carbon or in combination of 2, 4, 8, and 15. And then I had a condition in which all the resources were provided together. And across all these conditions, I kept the total concentration of carbon constant. So here, each of these carbon sources was 1/16, represented 1/16 the fraction of, this was the fraction of the total carbon. And then I used a daily dilution approach. So basically every day, I was taking a small amount of the culture and inoculated into fresh media. And I did this for 7 days, and at the end of these 7 days, I extracted the DNA and measured the composition of the communities using 16S amplicon sequencing. And now I'm going to show you the results of this experiment. And here I'm using this kind of plot as a canvas, where you have the number of supplied resources on the x-axis and the richness measured as number of amplicon sequence variants on the y-axis. And here, I put as a reference the prediction that would come from the competitive exclusion principle, which states that the number of coexisting species should never exceed the number of available resources. And here I'm starting to populate this plot, and these are the average richness of the communities grown in the different carbon sources, and here each color represents one of the specific carbon sources. And first you might notice that definitely it's not just one species that coexisted, and actually the average is pretty high, it's about 20 species. But also there is a lot of variation because, for example, these organic acids sustained only a dozen of species, while like sorbitol and cellulose were able to sustain up to 40 species. And here, one can start to speculate about the mechanism, but one that comes to mind is cross-feeding. That is this process by which microbes take up resources and then can leak in the environment the metabolic byproducts that can become food for other community members. So now I'm going to plot the what happened when we combine two resources. And our expectation is that, well, we had so much diversity in one resource, maybe when we combine them, we see a peak in diversity. Surprise? No. Actually, what we see is that when we combine these resources in pairs, we only see, basically, we don't see an increase in richness. And actually, the increase in richness that we see later is also quite weak, but definitely linear. So basically, for every resource that we add, we are basically adding one or two more species, which is kind of similar to what would you predict from the competitive exclusion, apart from this offset that might be related to cross-feeding. So to try to kind of unpack a bit this cross-feeding mechanism, we decided to go back to single resources and think about how we can quantify these metabolites. So just as a comment, I got this data mid-2020. So everything that could have been done experimental, like metabolomics, was kind of, okay, canceled, no thank you. And so we kind of got creative. And so this is a map showing well, you can recognize the central carbon metabolism and so with the glycolysis and the TCA cycle. And this is kind of a map that you can draw from KEGG, that is this database that contains many metabolic pathways. And here I placed this is a simplified map, of course. Basically, I colored where each of the supplied resource would enter in the map. And we use this map to do what is called a scope expansion analysis. So basically what you do is that you say, okay, we have a general map of the metabolism that is representative of the metabolism that can happen in soil communities. So exclude like pathways that are only present in marine species, for example. And then you ask, okay, if I have the resource that I provide in the media, like for example, sucrose, and some currency molecules like acetyl-CoA, ATP, water, then you ask, okay, how many pathways can I open, how many reactions can I open provided these constraints? And so what you get is basically a prediction of the number of metabolites that are produced based on these constraints. And of course, it's a very crude estimate because here we are agnostic to what is actually leaked in the cells by the cell, outside of the cells. So it's just how many metabolites you could produce in principle. But despite being very crude, I think when we plot it against the richness in single resources, we found a kind of striking correlations, meaning that those resources that were only able to sustain a few species, actually, you can only produce not so many metabolites compared to, for example, other resources from which microbes could produce many metabolites and so could sustain many more species. And this worked much better, for example, with other ways we could kind of estimate the complexity of molecules. For example, the molecular weight. Because you can see that where are the disaccharides, which supposedly will give more metabolites because they are bigger molecules. They're not giving more metabolites than, for example, I don't know, fructose in this case. No, this red here is glucose. Okay, so then we started asking, can we use this metabolic map to try to understand the linear relationship that we observe between the number of carbon sources and richness? And well, we used, again, an in silico approach, and specifically a consumer resource model. So in this consumer resource models, basically you have dynamics for the species and dynamics for the resources. And if you introduce cross-feeding, you also have a matrix that is telling you, for each species, which resource is going to consume and which metabolites is going to leak. So you have a correspondence between which metabolites are available and which species are going to consume them. Usually, this matrix is populated at random. Instead, our idea was to actually use this metabolic network to populate this matrix. And this works as follows. We have to make a kind of a big assumption, that is that all species preferentially consume one resource, and then they leak in the environment the two metabolic byproducts that are adjacent to the supplied resource in this map. So in this case, it's sucrose, and you produce this and this one. Then there are other species that consume these metabolites and release in the environment other metabolic byproducts following this kind of chain. And as you can see, when you start consuming, you will only leak a fraction of the original provided resource. And so you arrive at the point in which the metabolites are a very small concentration that cannot support growth. With this kind of construction, and we run the simulations of this consumer resource model, and we find that this model is able to actually recapitulate pretty well the linear relationship between number of supplied resources and richness. And the slope is, so if this one, if the observed one was 1.4, this is less, it's kind of 1.9 or 2. So it's pretty good. There is a decent agreement. Okay. Now, I said we manipulated the number and type of resources. At the beginning, I said, what about, I also introduced concentration. And one thing that you can ask is, okay, what is this model predicting if we increase the concentration of this supplied resource? Now, to try to understand it, we can simplify a bit this metabolic chain. Let's imagine that this is the resource that is provided. And again, here you have one species that consumes it and leaks it. And then you have only a chain, OK? And basically, here the concentration threshold at which the produced metabolite is not enough to support one species. In this model, if you increase the concentration of the resource, you basically make this chain longer. So the number of metabolites that are above the concentration threshold is just larger. So basically, this model makes a very clear prediction of what would happen if you increase the concentration of the resource, that you should see more like richer communities. Okay? So, well, yeah, okay. This is clear. So then we went ahead and we did experiments. So I started again with a pool of 12 carbon sources, and you can see some of them kind of overlap with the previous experiment. And we supplied these carbon sources, changing their concentration over 3 orders of magnitude. And then we inoculated this 96 well plate with this different concentration with the usual soil suspension with several microbes. And we did the dilution protocol and finally got the composition using 16S. And what do you think we saw? Okay, we saw a decrease in diversity. So basically, we tried all these different carbon sources, but what we see is a general, for sure is not an increase in diversity, and the richness of this community, so this is a number of ASVs, again, against the concentration as weight per volume. And we see that basically there is a slight decline in the number of species. Okay, this result is unpublished. We're still working because we're fighting to understand how to predict the decrease in richness with a consumer resource model. And apparently, the missing ingredient, and we discovered it by doing some spent media experiment, is that it seems that when bacteria have more resources, they can engage in more warfare. So the spent media becomes more toxic. And actually, when you introduce this trick in the model, finally it kind of reproduces this behavior and also keeps the linear relationship that you observed in the first experiment. But this is a lot of work in progress because we're trying to characterize more, and this is what my lab is going to do, hopefully soon, trying to characterize more these metabolites that are produced and are becoming toxic as you increase the concentration. Okay. Also, I'm used to take questions even during the talk, so if you have questions, please feel free to ask. Otherwise, we keep them for the end. Okay, so what we learned up to now is that it seems that we can find simple linear relationships between properties of nutrient pools and community diversity. So these are pretty simple linear, and hopefully they can be generalized to different systems. This remains to be seen. But also I think that what these studies show is that these bacterial microcosms can be powerful tools to establish causality between changes in the environment and changes in communities. And the other thing that I think is kind of exciting is that these results suggest that in principle we can modulate the diversity and structure of communities by tuning nutrient availability. And I can talk more about the structure because I didn't go over it at all, but in principle, I have a few slides. Okay. How am I doing with time? Okay, good. Let's switch gears and go to the second part about temperature. And so here we basically asked if we can find some general patterns in the way temperature structures bacterioplankton communities in marine environments. So what I'm going to show you is that we started from this result that increasing temperature favors low growers in pairwise competitions. And then we went ahead and showed that in the oceans, lower growing bacteria are generally more abundant where and when temperatures are higher. And also, what I'm going to show you is that temperature favoring slow growers seems a general pattern that applies to ecosystems beyond the ocean. Okay, so first, why should we care about temperature? Well, temperature is a cardinal variable for microbial life. And both the growth rate, like enzyme kinetics and bacterial growth rate can be very well described by what is called the thermal performance curve. And this is pretty well conserved across organisms. So temperature increases growth increases with temperature, and then past the optimum is sharply decreases because proteins start to have troubles folding properly, and then death ensues. But also, microbes are one of the few life forms on Earth that can really span the temperature gradients that we see on this planet. So they go from freezing temperatures to boiling temperatures. And so they're also classified based on that. But many of the studies thinking about how temperature affects community structure base their predictions solely on this. So basically, what are the permissible ranges of temperature where a species can live? And then you can predict, okay, I will find these species there. But what about interactions? And is there a way that temperature affects the outcome of interactions? So a couple of years ago, my colleagues published a paper actually looking at this specific effect. So what they were doing, they were competing these two species that, for simplicity here, are red bug and blue bug, at 3 different temperatures, 16 degrees, 25 degrees, and 30 degrees. And what they discovered is that across all this range of temperatures, these two species maintained a clear growth hierarchy. So the red species was always a fast grower compared to the blue species at each of these temperatures. But then when they looked at the outcome of competition, what they found that at 16 degrees, the red fast-growing species was winning. At 25 degrees, they observed coexistence. But at 30 degrees, surprisingly, it was the blue, slow-growing bug that was outcompeting the fast grower. And you can say, okay, maybe it was just a particular instance. But no, they tried about 40 pairs of species, and this result was kind of general. And they could explain this result using a Lotka-Volterra model in which growth rate is dependent on temperature. And I'm happy to go over it in the Q&A. But because here the most important thing for me was that this generates a very nice prediction that we can test if we have data about bacterial communities changing over temperature gradients. And we actually have this data for ocean microbiomes. And so the hypothesis was that slower growing taxa should be more abundant, basically where and where temperatures are higher. So in summer, at the equator, and the surface of the ocean. So with this with this hypothesis, Claire and I, that is my collaborator in this paper, and she also started the position at NYU, Claire Bello. So this paper was lucky for us, I would say. So we started this again was during the pandemic, so we're still working with data, basically. We started collecting microbiome data sets that were recording changes in bacterial communities across seasons, latitudes, and depth. So, and this is a map showing you 3 of the 7 data sets we've been working with. So one is this time series in the Baltic Sea. This time series is pretty amazing. At the Linnaeus Microbial Observatory, they've been taking samples of marine communities every 2 weeks or a month across 11 years, and this time series is still going. And they're also collecting temperature and other environmental variables, including dissolved organic carbon and other nutrients. And then we got our hands on this very nice transect. This is a cruise across the Atlantic. So this data set spans literally 100 degrees of latitude, so a huge variation in temperature. And also it has a bit of variation in depth. So these communities have been sampled in the photic zone between 0 and 200 meters. And finally, we also were able to use the data from Tara Ocean. Is anyone here familiar with Tara Ocean? Yeah, nice. Usually when I ask this in other departments, I'm like, no idea. Okay, anyway, so yes, so this data set, sorry. This data set is great because it spans like latitudes and longitudes, but also samples have been collected from the surface to 1,000 meters below, down the water column. And this set of data sets represents variation across 3 important axes of variation in temperature that are time and space, latitude, but also depth. And incidentally, this data is actually unpublished, so people that collected the data are actually co-authors in our paper. Okay, so, but what I told you before is that all our studies and our hypothesis is based on growth rates. And in the lab, we can grow our species in our plate reader, estimate maximum growth rate, and we can actually say, okay, this is the fast grower and the slow grower. So here, we had to resort to genomic proxies for growth rate. And this paper that came out in 2016 showed that maximum growth rate is actually proportional to the number of copies of the 16S RNA operon. And so basically what allows us to say is like we can make relative comparisons between species that differ by their copy number. So for example, this red bug that has copy number 3, we can say that potentially is a faster grower compared to the blue bug that has only one copy of the 16S RNA operon. But actually, if we have a community, we can calculate a mean copy number. So we can get the average of the copy number of all the species and weight it by the abundance of each species. And this synthetic observable basically gives us the abundance distribution of fast and slow growers in a community. And this has been used by other people, not just us, and it has been shown that responds to changes in the environment. For example, these studies were changing nutrients, and you can see that copy number responds to changes in nutrient availability. Okay, so now we had this way to calculate the abundance distribution of fast and slow growers, and we did it for all the sampling stations and sampling points, sampling times of each data set, and basically see their correlation. So I want to show you this data separately before. So this is how temperature varies in these three data sets. So no surprises here, at least. So in the Baltic Sea, you see, well, it's what you expect: cold in summer, hot in winter. Cold in winter, sorry, so hot in summer. And for the Atlantic, we have a peak in temperature around the equator and the tropics. And in the Tara Ocean data, specifically a slice of data where we had a lot of measurements along the water column, we see that temperature follows the typical, it's what you expect, and tends to decline as you go down the water column. Now below, I'm plotting the variation in the copy number. And you can see that across this data set, despite the variation that you might expect from in natural systems, you see that generally copy number values are lower in correspondence to higher temperature values. And so you can also visualize it in a nice correlation where you have temperature on the x-axis and the mean copy number on the y-axis. You can see that all three data sets show a negative relationship. But the other thing that I think is kind of interesting, and not exactly what I was expecting, that the slope are really similar. So across the three data sets, so very different basins, temperature changes across different environmental gradients, the slope is kind of the same. What is different is the intercept, and might reflect whether you have actually differences in the overall abundance of fast and slow growers. For example, you see that the Baltic Sea actually has a higher copy number, and I don't know, it might be because it's a very brackish system and because there is a lot of nutrient input as a possibility. But of course you can tell me temperature is not the only thing that is changing. So it might be, this result might be spurious, might be related to, for example, other environmental variables. So here we go back again to our time series, the LMO time series. And for this data set, we have a lot of environmental data because we have DOC, nitrate, ammonium, and phosphate, and other variables. But you can see that also these variables show some level of seasonality that might cause changes in copy number. So what we did a statistical analysis using these generalized additive models where we were basically able to separate the effect of temperature on copy number and from the effect of the other environmental variables on copy number, and take into account also like variations due to time and space itself. And these are the results. So here I'm plotting for each tile, basically represent the value of the parametric coefficient multiplied by the standard deviation of the environmental variables so that you can actually compare the coefficients. And cold colors, negative effects, red colors, warmer colors, positive effect, and the statistical significance is also provided. And you can see that even when you take into account other variables, and these variables can have significant effects, the effect of temperature is still there and still negative. But we also, I told you that we had other data sets. So here I'm plotting two other data sets. For example, this one is a time series in the Mediterranean Sea, where actually the temperature gradient was much smaller. So we have a smaller effect, still negative, even when you have a strong effect from the length of the day, so how much light these communities get. And also for this other time series, you still get, let's say, a negative effect of temperature. Of course, it is not very satisfying, this plot, because many data sets actually lack environmental data or were not available. But still, the LMO data and others give some confidence that the temperature effect is there and it's negative. Okay, last bit about this story is that we really think that this effect of temperature on the distribution of fast and slow grower is kind of general. So first, another example of marine environments. These are two maps showing you the distribution of Prochlorococcus and Synechococcus. And this is well known, so you have Prochlorococcus that is more abundant across the equator, and Synechococcus distribution instead extends toward temperate and polar regions. And incidentally, Prochlorococcus is a slower grower compared to Synechococcus. So actually, our prediction would be consistent with the observations that people make about Prochlorococcus and Synechococcus. Then the other thing is that we can look at soil experiments. So for example, this is an amazing experiment that has been going on in the Harvard forest, 20 years of warming of plots. And you can see that the control plots that were not warmed have a much higher copy number compared to the heated plots. So basically, again, our results are kind of consistent with what other people see in other environments. Okay, so what I want you to remember about this particular part of the talk is that I think we discovered a macroecological pattern in the distribution of fast and slow growers in marine communities that seems also that can be generalized to other systems. And to me, the most kind of mind-blowing things here is that really what this approach of using these experiments to generate hypotheses kind of worked. So, and especially when we have this overwhelming amount of data that we are generating, having some hypotheses to try to parse the complexity might be a good way to think about this data, even though these hypotheses might come from these very simplified experiments. And the final thing is that somehow we can also provide a prediction for how bacterial communities might change with warming, and specifically if, of course, if species remain within their optimum. Of course, if temperatures start passing the optimum, things might become different. Okay, the other thing that is not written here is that we have simple predictions also for how salinity might work. So with the same type of thinking, we have some models, local Volterra models, predicting that an increase in salinity would favor faster growers. And so with the postdoc in the Gore lab when I was still there, we started doing experiments with salinity, and we're actually kind of finding that this prediction holds even when you change environmental variable. Okay, so before time for questions, I want to give you more food for thought. So that's what is in my mind, at least lately, is that I think that general rules in microbial community ecology can be identified. There is some hope. And I think the challenge is actually finding the right level of coarse graining, or at which scale we should look for patterns. And this is also related to the question of which traits should we think about. For example, for us, looking at growth was a good way to find patterns, but there might be other traits that in other contexts might be more relevant. And I think the challenge is actually finding when each trait might be important in which context. Also, I think that this kind of bridging approaches is a good recipe to find and test these rules. Of course, and in general, I think that the more interdisciplinary we get, the better it is. And finally, that I think these studies also give some hope that at a certain point we will be able to get to kind of develop a sort of ecological toolbox for microbiome manipulation that can kind of side all the genetic manipulations that people are already performing. Okay, I think I'm okay with time. So I want to thank all the people that worked and are working with me on some of the projects, including, well, my previous advisor, Jeff Gore, and Ijo is actually working with me on the concentration, more on the modeling side, together with Akshit, Jacopo, and Hyun. And these are my collaborators for the temperature paper. And I want to thank also my current lab members. I reached the number 2, which is great. And we'll be working on more nutrient-mediated interactions and community diversity. And also, we are interested in the compounded effects of salinity and temperature on physiology of marine bacteria and how this can scale up to understand how communities might assemble. And finally, we are really keen on understanding how the level of the predictability of the environment affect trait ecology and evolution. And if you are interested in these things, you can always get in touch with me. These are my contacts. And also, I need to thank my funding sources, MIT and now Yale. And also, thank you again for inviting me. And I'm happy to take any question. Sorry, you got the cue, but you can also shout. You got.
I was just kind of wondering, have you guys done any kind of cross-field references? Like, I was thinking when you said this, thinking about the marine environment, the richness of this. I was doing a paper with a doctor named Amerland that goes here. There's essentially, if you have a diet that's a combination of the Mediterranean diet, the ketogenic diet, then you get a very rich diet, in other words. You don't get a very great variety in the gut microbiome.
Okay.
But you get this preponderance of a group called Akkermansia eosinophylla.
Okay.
It actually has a great positive benefit on human health because the metabolites that it makes are ones the human body can use.
Okay.
You know, I just thought of that because, again, that combines just about everything you said. You increase the richness, you'd expect to see more diversity, you see less. You see an improvement in health. This also might relate to the increasing warmth because 98.6 human body, right?
Okay.
You can see what the effects of it would be when we study that. But again, I was just throwing it out there.
Okay, so I should read more about these papers with the gut microbiome then.
But again, Mediterranean and keto diet, the low carbs, the extreme richness of diet, we have it on seafood, plant heavy, and you see nearly exactly what you saw here.
Okay, cool. Thank you.
So I just wanted to clarify something really interesting, by the way.
Thank you.
When you call something a faster grower, is it still a faster grower in isolation at the higher temperature?
Yeah.
So is there a general mechanism for why the slower grower outcompetes the fast grower when together but not in isolation?
Okay, I'm going to extract the math. Okay, we don't have a specific biological mechanism, but we understand it from like this Lotka-Volterra model, where we assume that the two species are competing. We are probably competing for nutrients, or we are agnostic to the type of interaction. But so basically, the model that we're using, this is just the equation for the slow grower. I'm here just with two species. So it's a classic Lotka-Volterra model where you have the per capita growth rate, and then you have the slow grower maximum growth rate, the self-inhibition term, and then you have the inhibition by the fast grower multiplying the population of the fast grower. And then we have mortality, because this model was originally developed for these daily dilution experiments. And you can absorb mortality and do some changes. And in the end, you can basically reparameterize the model, and then you get an expression for the competition coefficient that you see only depends on the ratio between mortality and growth rates. So how well you compete depends on whether your net growth rate. Your net growth rate, since there is mortality, also take into account the fact that you are dying, you are being diluted at a certain rate. And in these systems, mortality doesn't change with species or doesn't change with temperature because it is a dilution. Okay? And in these models, the competition coefficients, so the values of the competition coefficients basically tell you where you are in this space. So if both competition coefficients are larger than 1, you are in this bistable situation in which the winner depends on the initial fraction. Then you can have coexistence where both competition coefficients are less than 1. And then you have the fast grower winning or the slow grower winning depending on one or the other. Okay? Then in this model, what you do is basically say, OK, I put growth rate as a function of temperature. And you can do it in two ways. You can use Arrhenius, which is the classic form. Or there is another model that is taken from food science, that is the Rutkowski model. It has more parameters, same results, so we stuck with so we decided to stick with the Arrhenius. So basically, here you can, this is, I'm showing you what you were asking. So if you are assuming that you have a faster grower that is always fast, maybe because it has more copies of the copy number. So that's this R that you see here. So if you increase temperature, actually the difference in growth rate seems to be favoring the fast grower, okay? But then basically, but what happens is that this ratio becomes since it's a ratio and mortality stays the same, the growth rate of the fast grower actually increases faster, so actually it gets less favored as you increase temperature. So if you plot instead of this, I don't think I have it, unfortunately. But if you basically plot the ratio, you would see the opposite. So that the ratio for the mortality over R decreases much faster for the slow grower compared to the fast grower. So the two species actually can become kind of similar in terms of net growth rate. But this net growth rate basically multiplies the competition coefficients. So if you start from the fast grower winning, if you increase temperature, you end up with this slower growing winning. And then you can pass from the coexistence as they show in the other paper. And then what we did in the temperature paper, so yes, we expanded the Lotka-Volterra model to 100 species. So this is a generalized form of the Lotka-Volterra model. We kept mortality in saying, oh, in the ocean there might be, like, cells are dispersed, cells are grazed, are lysed, and we assume that the way you get these processes are not species dependent nor temperature dependent. And then you can have the same form of the growth rate depending on temperature. And in this particular condition, you can use a geometric distribution to pick the copy numbers. And why the geometric distribution has a biological meaning? Because when you look at the abundance distribution of copy numbers in the ocean, the peak, so the peak is 1, and so the P of the geometric distribution actually represents really well the mean copy number in the ocean. And that's how the simulation, so you let the communities equilibrate at different temperatures, and that's what you see. So basically, even this generalized Lotka-Volterra model has this trend. And here you can do kind of the same thing for the equilibrated communities, calculate the mean copy number. But the craziest thing, if I can, sorry, thank you for giving me the opportunity of making my talk longer, that we can fit the Lotka-Volterra model to the data, which was kind of impressive to me. So these are the real points, and the black line is the fit of the Lotka-Volterra